Welcome, everyone. Before we start, we wanted to say thank you for your participation today. We are well aware of how important everyone's time in lab is, and we appreciate your time away from it. So first up is a colleague of mine, Dr. Kristen Sakink. She's a senior scientist in scientific affairs here at Arima and holds a PhD in biology. At Arima, she spearheads a number of research and development initiatives, and she's got a couple of really fun clinical projects at the moment. Kristen, the floor is yours.
All right. It is my great pleasure to be here today to share some of our latest research from Arima Genomics. Today, I'm going to start off by giving a brief overview of what it is we mean when we talk about 3D genomics, before diving into an example of how Arima's technology, and particularly our Capture-High-C methodologies, can help disease research uncover new mechanisms across a range of disease states. So let's dive right in and start by defining what the 3D genome is. In order to understand what we mean when we talk about the 3D genome, let's start with what we're all maybe more familiar with, what we might call linear genomics or 1D genomics. And we can define linear genomics as this process of figuring out the order of each of the approximately 3 billion A's, G's, C's, and T's in the genome in one or a few contiguous sequences. But of course, there's far more complexity to the genome than can be easily captured in this linear paradigm. What we know now is that DNA is not just randomly shoved into the nucleus of cells. There is this defined pattern to how chromosomes are packaged into compartments. And this is what 3D genomics allows us to describe. 3D genomics takes into account not only the sequence, but also the structure of how the DNA is organized in the nucleus to better understand gene regulation and genome function. So when we think about 3D genomics, we can see that the sequence is really just the tip of the iceberg when it comes to understanding genome function. Using a 3D genomics approach, we can understand not only the sequence, but also structural variation, translocations, indels, inversions. And going further, it helps us to understand the mechanisms of gene regulation, or even broader genome organization like chromosomal compartments, topologically associating domains, or looping structures, all of which can affect genome function. And the types of questions that we can address with 3D genomics have applications in a lot of different areas of active research. 3D genomics allows us to describe the spatial organization of chromosomes and has contributed greatly to genome assembly, including the recent publication of the first telomere to telomere human genome. 3D genomics has also had an enormous impact on epigenetics as it captures how spatial and temporal changes in chromatin conformation can alter gene regulation and cellular function. But the real promise of 3D genomics is that we can leverage these structural and functional insights from the 3D genome and apply them to major challenges in human health, with the goal of being able to understand the mechanisms underlying disease and ultimately of developing new therapeutic approaches. So let's take a look at an example of how 3D genomics provides key insights to understand and treat cancer. So this is a really exciting paper that was published in Nature Genetics in 2020 by Cloken and colleagues. And what they were able to demonstrate here was how the 3D organization of an oncogene, in this case MYC, relative to its enhancer, could affect the progression of T-cell acute lymphoblastic leukemia. So in this first set of diagrams representing a healthy patient, we can see that the oncogene MYC and this putative enhancer element are approximately 2 megabases away from each other in linear space. And 3D genomics shows us that these two elements normally arrange into 2 individual loop structures that are very distinct from one another. However, in patients with this leukemia, we see new transcription factor binding activity at the enhancer and a corresponding activation of the MYC gene 2 megabases away. Taking a 3D genomics approach, we can see that there's a rearrangement of the chromatin loops, and this leads to the enhancer element being brought into close proximity to the MYC gene, causing the aberrant expression at MYC. The authors of this study went on to show that by inhibiting that transcription factor bound to this enhancer with pharmaceuticals, they were able to restore normal expression of MYC. So using 3D genomics, they were first able to identify the mechanisms by which MYC is misregulated in T-cell acute lymphoblastic leukemia. And secondly, to inform exactly which transcription factor needed to be targeted by the treatment to restore normal function. So now I want to shift gears a bit and talk in more detail about Arima's Hi-C technology and how it can be used to gain these sorts of really valuable insights into genome function. The core Arima Hi-C platform has a few different capabilities. Genome-wide Hi-C and high-coverage Hi-C, as the name implies, allow the exploration of these 3D interactions across the entire genome in an unbiased manner. But we also offer methods to selectively target different genomic regions of interest. Hi-chip essentially combines ChIP-seq with our core Hi-C technology to enable the detection of 3D genomic interactions that are associated with the binding sites of DNA-associated proteins. Capture Hi-C then similarly uses probes to capture and enrich for genomic regions of interest in a Hi-C library. Arima offers a couple different options for Capture Hi-C. The promoter capture panel offers a comprehensive probe design targeting promoters across the entire genome of human or mouse. But we also offer custom capture panels that can be tailored to a specific research question. For example, targeting regions surrounding non-coding GWAS variants or other larger regions of interest like genes or exons to address specific hypotheses. So why might you want to use a capture-based approach to Hi-C? Well, Capture Hi-C can help us to understand long-range gene regulatory mechanisms, and it can help facilitate testing hypotheses that are formulated from other genome-wide DNA analyses. But Capture Hi-C really can facilitate that by enabling us to have increased resolution for identifying those 3D genomic interactions across our regions of interest, while also reducing the overall cost and increasing the computational efficiency of the analyses downstream. So to illustrate how we're able to achieve these benefits using this Capture Hi-C approach, the top section of this figure here depicts long-range 3D interactions in the region surrounding this example gene that are identified using genome-wide Hi-C. As you can see in the middle panel then, only a small fraction of these genome-wide Hi-C interactions actually involve the PRR5L promoter, the part that we're interested in here. But by using a capture panel to enrich for promoter regions, including the promoter of this gene, we are able to effectively increase our sequence coverage of that region that we care about, the promoter region, as we can see in the bottom panel here, making it much easier to detect the significant interactions between the promoter and different enhancer elements at a much higher resolution and with far less total sequencing. So the workflow for either promoter capture or custom capture Hi-C can be divided into essentially 4 different main components. The first step is the sample preparation using the Arima Hi-C kit. I don't have time to talk about the details of the protocol today, but the important thing to note here is that this module allows us to isolate the DNA while retaining that all-important 3D information that we're interested in. The samples are then ready to be used in standard NGS library prep protocols, and those resulting NGS libraries are enriched for the regions of interest using our capture probe set. And enriched capture libraries are then ready for sort of the third step of this workflow and can be sequenced on the platform of your choice. And the resulting sequence data can then be analyzed using our open source bioinformatic pipelines. So overall, the Capture Hi-C workflow will work with a range of different sample types. It is really a very user-friendly workflow that produces robust and reliable results. So to finish up my portion today, I'd like to walk through an example of how promoter capture Hi-C has been used to help understand disease mechanisms and inform therapeutic approaches in cancer. But the example that I'm about to share is, of course, only one of many recent examples of very exciting studies from diverse fields that have used Capture Hi-C to gain valuable insights into the mechanisms of disease. And we will also hear more about one of these examples shortly from Yoshi today. So the research that I will talk about today was done in collaboration with Joanna Ockinger-Kawaka at the Garvin Institute. And the goal here was really to understand the changes in gene expression and the regulatory landscape of breast cancer tumors using a multi-omics approach. So to do this, they grafted patient cancer cells into a mouse model where they were treated with the demethylation drug decitabine or with a saline control. And after treatment, they performed a lot of different assays to measure DNA methylation, gene expression, transcription factor binding. And of course, what we're interested in here is 3D genomic structure with genome-wide Hi-C and promoter-anchored 3D interactions using promoter capture Hi-C. And I'll be focusing on these last two in the last couple of slides here. So what I'm showing here are the results of the genome-wide Hi-C, which really gives a broad overview of the genome structure and the chromosomal domains in each treatment. And if we compare the region marked by the asterisks here in samples that were treated with the drug in the bottom panel versus those that were not, there is a significant change in the 3D conformation of the genome, the loss of this topologically associating domain, or TAD, at this location in the treated samples. Now, what we think is happening here is that the drug decitabine is inhibiting DNA methyltransferase, causing the hypomethylation of the DNA in the treated samples. And the loss of this methylation leads to decompaction of the chromatin and subsequent changes in the 3D conformation, which in turn affects the expression of genes in that region. So the authors here went further to test this model using promoter capture Hi-C, where they could just focus on the interactions between gene promoters and their enhancers to see how they might affect gene expression. And this figure shows the results for just one gene, KRT8. KRT8 is a member of the keratin gene family, and misregulation is known to promote metastasis in some cancers. So here you can see that in the decitabine treated cells, there has been this gain of multiple long-range interactions between the promoter here and putative enhancer elements. They also saw similar patterns in other key genes, sort of supporting this model that many enhancer sites were being demethylated and reactivated following treatment with decitabine, which they also showed led to the activation of estrogen receptor target genes and tumor suppression pathways that are associated with good patient prognosis in these endocrine-resistant estrogen receptor positive breast cancer patients. So in conclusion, I hope I've helped convince you that there is much more to the genome than just sequence. And by using this 3D genomics approach, we can really provide novel insights for disease researchers. I hope I've also convinced you that Arima's Hi-C technology and the capture Hi-C technology in particular are robust and reliable solutions that can give very valuable novel insights into disease mechanisms. So finally, if you think that either genome-wide Hi-C or capture Hi-C is something that you'd be interested in trying in your own lab, Arima provides both kits and end-to-end services and support. So feel free to check out our website or get in touch with us to discuss how 3D genomics might benefit your research. And with that, thank you all for your attention. I will turn it back over to Christian to introduce the next speaker. Thank you.
Perfect. Thank you, Kristen. Just wanted to introduce Dr. Yoshihiro Izumiya. He's a molecular virologist at my alma mater, UC Davis, and focuses on the molecular mechanisms of Kaposi's sarcoma-associated herpesvirus reactivation from latently infected cells. Dr. Izumiya utilizes viral mini-chromatin and viral host interaction and entry to understand cellular protein functions. Thanks for joining us, Yoshi, all the way from UC Davis. Really appreciate it. The floor is yours.
Okay. So thank you so much for giving me the opportunity to talk here today. So today I want to talk about how we can utilize the 3D genomics to study the viral gene expression like regulation. So the first, why we are interested in the virology? The first foremost is the virus is extremely old. Therefore, it's some retrovirus is 450 million years old, likely originate for the billion years ago before the cell. So the virology is a study of the genetic evolution. And then also our genome, when looking at our genome, the many viral genome is encoded in our genome. It's like, and then the protein encoding region only 1.5% per viral genome about occupy the 8.3% of the genome. And viral particle are extremely beautiful, and then the evolution, beauty of the evolution. So the big picture of the biology in my mind is that the integrative science with the study of the molecular wizard, which is a virus. So that, for example, 2017, it's only the 1 year we about 1,000 of the viruses identified. So virus is everywhere in our world. And also, for example, there is about 10, 16 of the HIV genome in the planet today, and more than 1 liter of the coastal seawater has more than the people on Earth. So if you buy that 100 centimeter square, the material leaf, the organic spinach, for example, you can identify 10 to 8 of the viral particle in there. So the diversity and the constant evolution makes this molecular wizard virus very special. So viruses are very smart with a million of the years of the accumulated knowledge about our host cell, and because they need to replicate and utilize the host cell to replicate, so they have a lot of knowledge accumulated, and then we can still learn a lot from them. So our lab is working on the KSHV. The KSHV is the Kaposi sarcoma-associated herpesvirus, the gamma herpesvirus. The KSHV is also known as the HHV8 because it's identified number 8 of the human herpesvirus. The virus is originated detected in the KS, Kaposi sarcoma region, 1994. That has since been observed the 95% of all the KS tumor. That this virus also associated with the other B-cell lymphoma, the primary effusion lymphoma, which you can see here is a lymphoma. And when looking at the viral protein, one of the protein called the RANA protein, you can identify where the viral genome is located. And this green dot is where virus genome is located. So the other, as similar to the other herpesvirus, the KSHV also have the 2 life cycle, one called the lytic replication, which produce the viral virion particle and then infect to another neighboring cell. And here is a latent infection, just staying as a dormant, and then doesn't express much of the protein, except a very few protein, and then stay latent to hide from the host immune system. So the one characteristic of the KSHV is the one viral protein, which we call the KRTA protein, which is a very strong trans-activator protein, the transcription factor. Overexpressing this KRTA into that latent infected cell, we can induce that the virus lytic gene expression. So we utilize this system to study how genomic structure change and how gene expression is regulated. So we think this is very exciting model system to study the gene regulation in general. I'm going to show you why. So first, the KSHV genome is very small compared to the human herpes. The human genome is only about 140 kilobase pair. So compared to the cellular gene, for example, PRB, which is spanning the 200 kilobase pair. So another thing is that in one cell, this is one cell, has the multiple viral genome, identical mini chromosome viral genome in the one cell. So very easy to do the chip sequence because you don't really need to do that enrich and have the very good signal, and then we can do the RNA sequence. So then another thing is that this histone, this virus chromatin, the episome is also modified as similar to the human chromosome. These are the cut and run study with the show the sum of the active histone mark and the repressive mark. As you can see, these are condensed region has to have the methylation of the H3K27. And also you see the open chromatin region has the active histone mark. It's very nicely decorated. So then another exciting advantage is that we can have the back recombination system. What is the back recombination system is that the entire KSHV genome is in the bacteria. And then we can manipulate the viral genome at the single nucleotide resolution inside the bacteria chromosome, inside the bacteria. So what it does is that with the KSHV back system, the viral genome will become manipulatable, the mini chromosome at the single nucleotide level to study the very basic gene regulatory mechanism, which is also equipped with the on or the off switch. So it means that the KRTA inducible cell. So for example, induce the KRTA with the doxycycline inducible promoter, we can induce the entire KSHV gene expression so that we manipulate the genome, turn on the KRTA, and then we looking at the virus gene expression as a reader. So this is a very good convenient system to study the gene regulation. So with this system, we are interested in 2 things. I want to show you the 2 things. One is the function of the non-coding RNA in general gene regulation. And also I want to show the example that we can utilize this viral protein sequence, the protein-protein interaction as a starting point to generate the cancer drug. So the quick introduction of the non-coding RNA, as you may know, that the case our genome encoding the tons of the non-coding RNA. Some is a small, some is a longer. So what the definition of the long is that 200 base pair is more than the 200 base pair is we call the long non-coding RNA. So large number of the non-coding RNAs are encoding our genome. And then the previous study also showed that when looking at the non-coding RNA sequence itself, it's not really the conserved among the species, but the position is conserved between the species. So then we are interested in what the non-coding function is. So as similar to our human genome, the KSHV and also the large double-strand herpesvirus also encoding that the non-coding RNA. Here is showing the chicken alpha herpesvirus, showing that these are the non-coding RNAs expressed from the viral genome. Here is showing the gamma herpesvirus, the Kaposi sarcoma herpesvirus. So during the replication phase, there is a huge amount of the non-coding RNA expressed from the KSHV genome. So the question is that what's the advantage for the virus to keep long non-coding RNA in its genome and expressing 100 to 1,000 times more of the open reading frame. So these are open reading frame and the non-coding RNA express way much more than the protein coding sequence. So my main question is that evolutionarily maintain this long non-coding RNA for the million of the years in very limited genomic space. It must have very important reason for what it is. So then to study this, first we mutated with the promoter region of the non-coding RNA so that we inhibit the non-coding RNA expression from the KSHV genome. Then asks the question, what's happened? So by mutating the promoter region, we completely abolished that non-coding RNA expression, but in the same time, we see that many other genomic regions in the domain-specific manner reduce the gene expression. Then we mutated this non-coding RNA. Then we found it's very similar but a different domain. This gene expression is down. This suggesting that the link RNA expression regulates the distantly located promoter activation. But how does transcription activity have effect on the distal gene expression? So then it's come down to the 3D genomic structure question. So to study the 3D genomic question, so we buy the Arima kit to do some ligation, and we also enrich the KSHV genome before sequencing, because the KSHV genome is only 150 kilobase pair, but while the human genome is 3.2 gigabase pair. If you just simply sequence it, we're going to sequence too much of the human genome. It's not cost effective. Then we ask the question, what the genomic structure is. So here is a show in that the heat map of the 3D structure and that the genomic frequency of the genomic ligation. So as probably you know that these red regions showing that if these two fragments is interacting more frequently than that theoretically expected, you see these red. And for example, these regions also have the genomic looping. So then we compare with three different naturally infected KSHV cell line, B cell line, the primary effusion lymphoma cell line, and then ask if the KSHV genome structure is conserved. So as you can see, these three different cell lines isolated from the different patients show that the very similar genomic structure. And then here is showing the experimentally infected cell. But while we directly compare between these two cell lines, we start to see the difference. So this suggesting that the overall position to have the genomic contact are conserved, but the frequency from the different domain are different among the cell line. So how the border of the genomic group is coordinated in the KSHV genome. And next, we performed several cut and run study to supplement with the genomic study. So as you can see here, the CTCF cohesion located the border the corner of this triangle and seems to be responsible for the genomic interaction, which is very similar to the host genome regulation. And the position of the RNA pol 2, the post RNA pol 2 are located at the border with the CTCF, which is situated very next to the viral transactivation activator, KRTA binding site. These are the KRTA binding site located very next to the CTCF and very next to the RNA polymerase 2, post RNA polymerase 2. The genomic region encoding the viral form the frequent genomic group. These are the where the coding RNA express has the frequent genomic grouping. So next, based on this frequency of the genomic grouping, we generate the 3D genomic models. So here is showing the during the latency, how KSHV genomes looks like in the 3D modeling. And here is showing the after induction of the KSHV gene genomic structure. As you can see, these are really the sphere shape, but once induced the KSHV gene expression, it becomes more like a donut. And then we also see that this non-coding RNA, which is expressed about 1,000-fold more, is more moving towards to the inner of this donut, showing that RNA polymerase 2 may be recycled and utilized for the KSHV gene expression. So we call the KSHV genome structure the bird cage model, and we recently published the Journal of Biology. So then the fundamental question is still remain, and then we kind of know that the transcription is shaping that the gene structure, and this genomic structure is shaping that the transcription, the richness of the specific gene expression based on the promoter and the enhancer interaction, and these determine the tissue-specific genomic structure. So these are general idea, and then I think we accepted that, but it's very difficult to study. So to do that, to study this, we next study that if we inhibit the initial viral gene expression before establishment of the latency genomic structure, will the virus genomic have the different genomic structure so that it becomes non-deactivatable? So if so, isn't it the molecular mechanism of the non-permissive interaction? So that what we did is we first using the back system to mutate it that some transactivation protein expression, then study the genomic structure, the looping. So we using the back system to mutate the inserted the stop codon for the one of the transcription regulator of the KSHV, and then we generated the recombinant virus and then infected this virus to study what the genomic structure looks like. So here is showing the wild type, the virus genomic structure. So we see like this. Then while we mutated that, what we see is that some segment of that genomic domain start to losing that genomic interaction. So especially that the linked non-coding RNA encoding region losing that genomic contact with the anagenomic region. And then we next study to see how does it translate the gene expression. We performed the PCR array to study all of the 84 viral gene openly linked claim expression. So what we see that essentially all the viral gene expressions going down, especially the linked RNA expression. So this tells that the less transcription leads to the less genomic structure, less long range genomic grouping in the latently infected chromatin, and then less reactivation potency. So less genomic grouping leads to the less gene expression and then abortive infection. So that we talk about the within the KSHV genome the genomic interaction. So next, I want to show you about how KSHV episome, where KSHV episome are tethering on the host chromosome. So here we study the virus genomic interaction. Next, we study the virus genome and the host genome interaction by studying the chimeric sequence. So the KSHV tether the host chromosome through the runner protein. So KSHV episome is docked to the host chromosome. So our question is where is this and why they choose that region. So by studying the tethering site on the host chromosome, by studying the chimeric ligate sequence with the host chromosome, And then what the host proteins are involved, we perform the proximity biotin ligation for the runner protein to looking at the surrounding protein. So here is showing that the high-C data for the 3 different KSHV infected cells. So these each black dots indicating that the sequence read from the KSHV and then also the host chromosome chimeric sequence read. As you can see, here is a chromosome 1, here is a centromere region, you see some peak nearby. So this data was also confirmed by the DNA fish we stain with the centromere region with the DNA fish probe, and then also stain that the runner protein, which is important for the tethering to the host chromosome. As you can see, for example, blue and red are very next to each other for the many places. So then when looking at the G3, if this binding site is a random or not, we looking at the some Jacquard index. So what we see that the G3 cell line has a very similar position for the tethering site. For example, BC1, BC3, about 97% binds to the same site. So then here is a summary of the where's the KSHV episome tethering from chromosome 1 to Y. You can see these reds are the all 3 are located in the same site, and different colors show the some specific or some 2 are common. So these data suggest that the similarity of the KSHV episome docking sites among the 3 different cellularly naturally infected cancer cell lines suggests that there is a preferential nuclear microenvironment for the KSHV to stay dormant in the latent infection. So we performed the proximity biotin labeling to study what's the nuclear nanoenvironment at the KSHV tethering site. So what's the proximity biotin ligation? So this is a turbo ID. This is the biotin ligase. First, we fuse with the runner protein, which is important to tethering the host chromosome. And in the presence of the D-biotin in the tissue culture media, this ligase will biotinate in surrounding protein. Then we lyse the cell, then isolate with what the protein is biotinate, which is an indication of what the protein is nearby with the runner protein. Another advantage, the streptavidin biotin interaction is extremely tight, so we can watch the very harsh condition. So to do that, by doing so, we identify several proteins with the protein is nearby to the runner. So one of the protein, the CHD4, we performed all the siRNA screen to see which protein has a significant effect of the KSHV gene expression, and we identify CHD4. We then identify the protein complex in the purified complex, and we also confirmed by the co-localization, the immunofluorescence assay. So what's the CHD4 protein? So CHD4 protein is a subunit of the multi-subunit nucleosome remodeling and associated with the gene depression. CHD4 is also part of the heterochromatin, the KSHV complex, and that is involved with the gene depression. Frequently overexpressed in the many cancer cell types, and the mutation is associated with the cancer and the intellectual disability syndrome. CHD4 regulates enhancer accessibility and often localize fibrous region of the link RNA via positioning the CTCF. So then to study what's really the biological function of the CHD4 next, we first knock down the CHD4 from the cell, then we infect the KSHV, and then looking at how quickly KSHV can go into the latent state, the dormant state. So we perform the RNA qRT-PCR. So what we found that when knocking down the CHD4, KSHV cannot really enter to the latent state, the struggle to inhibit the gene expression, lytic gene expression. And then next, we looking at how the maintenance of the dormant KSHV. So we use the already latently infected cell, then induce the KRT transactivator expression inside the cell to deactivate the KSHV. In the same time, we put the mouse CHD or the cDNA to overexpress CHD4. So what we found is that here the control overexpression of the CHD4 significantly inhibits the KSHV deactivation. Then when knocking down the CHD4, you can enhance about a 20 times fold of the more reactivation. And then when put back into the mouse CHD4, and again inhibit the KSHV reactivation. So that the CHD4 is very critical for the KSHV reactivation. And then when looking at the single cell level, here is showing the KSHV non-express. KSHV is not the reactivating population. This has a more of the CHD4 expression, whereas the cell population that the KSHV gene expression is induced and expressed. Here you can see the CHD4 is not expressing well in this cell population. So these suggest that the KSHV does not establish the latent infection in the KSHV knockdown cell efficiently, and the overexpression of the exogenous CHD4 strongly abolishes the KSHV deactivation. So we recently reported this to the cell reports. So why so this CHD4 is so effective? So looking at where the CHD4 is actually locating on the KSHV genome, here is showing the cut and run study to locate where the CHD4 and runner location. What we see that this CHD4 runner locating in the promoter region of those highly expressed non-coding RNA region. So what we think is that the CHD4 is positioning the CTCF to inhibiting the elongation of the RNA polymerase to express the non-coding RNA, which inhibited the lytic gene expression. So because CHD4 runner is very tightly interacting, we next study that this interaction. So the KRT and runner and the CHD4 in vitro and the characterize on the chromatin. And then when we next utilizing the CHD4 and the runner interaction, and then try to identify if we can generate the inhibitor of the CHD4 by utilizing this interaction. So we map the where the runner interact with the CHD4, we map this genomic protein region, and then we further narrow down with the tithering biotin peptide, and we identify this region. And then we looking at the peptide and CHD4 interaction with the BLI, and we found that this very tightly interact. Then we fused with this peptide with the cell penetrating peptide to introduce this peptide into that cancer cell line. What we see is that by introduce this peptide into the cell, incubation CHD4 binding peptide kill that lymphoma, which is independent of the KSHV infection. So what we think is the overwhelming that this peptide to the binds the CHD4, inhibits the CHD4 function, which leads to the cancer cell death. We further confirm this xenograft mouse model. We see that this wild type peptide can inhibit the tumor growth in the mice. So another good thing is that this peptide itself does not kill the mice. So peptide identified from the evolutionary conserved viral protein sequence among the gamma herpesvirus inhibit the cancer cell growth in the mice. So then we think that using the viral host protein interaction as an entry point, we may be able to identify the cancer drug. So the key takeaway from this talk, so infected mini herpesvirus chromatin is similar, decorately similarly to the host chromosome. Therefore, it gives us a very nice model to study the gene regulation. So Hi-C technique generate a 3D viral chromatin structure model and then reveal the 3D genomic structure change which induced by during the gene expression. So Hi-C technology identify the preferential viral chromatin docking site on the host chromosome. The combination of the proteomics approach with the recombinant virus identify the T histone enzyme which is required for the maintain the KSHV episome as a dormant. The studying on the interaction between the viral and the host protein developed by during the evolution of the virus is a very exciting approach to develop the unique drug candidate.
Thanks, Yoshi. Really appreciate the talk. We'll have questions in just a moment. Just on my screen, you'll see something we're excited about over the course of the next few weeks, and we launched this back at the start of July. It's a disease research grant. We hold these grants on something of a quarterly basis, so ongoing through the month of July through August 5th. you have the opportunity to apply for a research grant. Let us know just a little bit about how you might use our technology in your lab, or potentially send your samples to our services lab where we can process them for you. We have some questions that have come up both in the chat and in the Q&A box. So just a reminder to pop those questions in the Q&A box at the bottom of your screen. We'll have both of the speakers today try to address those questions over the next 10 or 15 minutes. And I think we have one that just popped up, I think towards the end of Kristen's talk. Thanks again, Kristen, and thanks Yoshi for presenting today. Really appreciate it. I think the question might have come during your talk, Kristen, but I think probably all three of us could maybe tackle this question. So love to hear some different insights into kind of the comparisons of ATAC-seq and Hi-C technology. I know, like you presented, Yoshi, many of our customers are layering multi-omic information to answer different questions within the systems that they work within. So would love to hear your insights on maybe how you compare and when you might use and layer in ATAC-seq and or Hi-C.
Well, I guess I can jump in.
I can dive in too, but yeah, if you'd like to put your consent in, Kristen, please do.
Yeah, so this is a good question. I think the ATAC-seq and Hi-C, they're addressing slightly different questions, and they're complementary to each other, but certainly addressing different things. So ATAC-seq is addressing the accessibility of chromatin and how open or closed your chromatin state is. Whereas Hi-C, you're really looking at these long interactions between two pieces of DNA and whether those pieces of DNA have been brought into proximity by like protein interactions, mediating those. So they're certainly complementary, certainly both very important questions to address in epigenetics, and can work together really well to give you insights into what's going on in genome function.
Excellent. Anything to add, Yoshi?
Yeah, I agree with the question that yeah, it's a different question to ask, and the ATAC-seq is, we can also do some like a single cell level recently, so that's going to be very exciting approach to do that.
Excellent. Excellent. We have a couple of other questions here. I don't think they're in the Q&A box, so I will just read them out loud. Let's see, and they might be for both of you. So the first is promoter capture Hi-C. Can it also capture CTCF interactions? That might have come during your presentation, Yoshi. I know you're talking a bit about, yeah, again, layering of different techniques and technologies. Any insights there? And maybe Kristen, you can as well.
So maybe, so we are doing, we capturing with the KSHV genome, not the antibody to capture the CTCF. So because of the KSHV genome is smaller than the, much smaller than the host chromosome, we have to capture it in order to have like a significant depth to analyze. And then, yeah, we also recently started doing some of the capture study with another antibody. And then I think this really helps to increase the depth of the analysis and then the statistical point of view.
Okay, great. And I might suggest if you're interested in CTCF or some of the kind of antibody pull down work, there's also the high chip application a number of our customers have utilized. So combining Hi-C with ChIP-seq, just a little shout out for that application, but you know, promoter capture Hi-C and capture Hi-C in general can also be used to understand specific enhancer promoter interactions, et cetera, et cetera. Another question here I'll read out loud that I don't think everyone else can see. I think for you, Yoshi, we are working on viruses integrated into the genomes. With a viral genome of around 180 kb, do you think it is possible to form TADs within the viral genome, especially if there are CTCF binding sites?
Yeah, so the TAD is not necessarily the super big. It needs to be the really like a large like a size. We have done the KSHV, and then we recently published the Journal of Virology. Yes, it does form that the TAD formation, and each TAD is regulated or clustering with the different kinetics of the gene expression. So that we believe that it does form in the TAD, and this TAD does regulated separately from the different TAD. And then each TAD is separated by that cohesion as well as the CTCF, as very similar to that host chromosome. So yes, I believe that these also form that TAD.
Okay, excellent. I appreciate the insights. We'll keep it going with another question that popped up in the Q&A box. Again, just a reminder to use that at the bottom of your screen for any questions. We've got another 5 or 10 minutes here. Let's see. So how can we detect the interaction between virus genomes and the host genome if virus genome is integrated into the host genome?
How? I believe still you can identify some of the chimeric sequence, right? Maybe I misunderstood the question, but when you do that, those capture high C or the high C technology and the frequent ligation, we still see some like a preferentially ligated genomic fragment, which is indication of that this genomic region is more closer to the another genomic region.
Okay. Perfect. Yeah, I'm trying to reread the question here, but I think that makes sense. Kristen, another question we can't see, maybe just, I know it's kind of a common question. You might have touched on in your presentation. Can you just speak to maybe the sequencing depth required for some of the capture high C applications? I think that's something people think about genome-wide high C, and just the depth of sequencing that's required there can be obviously in the kind of mid hundreds of millions of reads, paired-end reads per sample. How does that compare or contrast to capture high C?
I think that depends a little bit on your question, what you want to get out of it, and like how big your probe set is as well, like how many regions you are looking at. So there's a bunch of considerations that need to be taken into account when you're deciding on the depth of coverage that you want. For what I work on a lot of like large structural variants in cancer with capture high C, and I get really good results from that with 10 or 20 million reads. Something where you're actually trying to look for loops and loop calling or enhancer elements in a smaller range, you might need more like 50 million reads. But yeah, it kind of depends on what your question is. So maybe that's we can work with you and try and figure out a good depth of coverage for your question. But yeah.
So in our case, at the 150 kilobase pair, we cover that the 5 million sequence leads to cover the 150 kilobase pair, which gives us about the 200 base pair average resolution. And then we generated the third calling in the 500 base pair resolution.
Perfect. Yeah, again, kind of your system of interest and maybe some of those technical questions that we can always help work with any one of you out there still online about your system of interest and potentially applying the technology. I know we have our human promoter capture high C kit, for example. We recommend starting with about 100 million paired end reads. So just again, kind of depends on the question that you're trying to answer. And again, please reach out to us and let us know if you have any questions about any of the technologies that Arima can help you with in the field. I'm looking at the Q&A box. I don't see any more questions coming through. Don't see any more questions in the chat. So I will just thank both of you, Kristen and Yoshi, for your time and presentation today. Really appreciate it. Again, great to see someone from UC Davis online here. So thanks for joining us. We do again have the grant, so please keep that in mind as you think about your research and some of the questions and maybe how high C technology from Arima can help. And wish you all a great day. Thank you again for your time and presentations. Cheers all.